This is TWIS, This Week in Science, episode number 483, recorded on Wednesday, October 1st, 2014. If you believed they put a man on the moon. Hey everyone, I'm Dr. Kiki, and this week on This Week in Science, we're going to fill your head with the man in the moon, magnetic dolphins, and a little wetness. But first...
Disclaimer, disclaimer, disclaimer. Time is a tricky thing. Time can fly, drag, march, run out, heal, and be killed. You can give it, get it, have it, take it, leave it, be ahead of it, be behind it, spend it, save it, lose it, and waste it. Once it's gone, you can't get it back. But you can always make more of it for the things that matter most. And as we learned from Einstein, it's all a matter of perspective, as time doesn't tick the same way everywhere for everything. So, while we begin our show at the same time of the day this week, but a different day of the week this time, we hope that the sometimes confounding nature of time doesn't keep you from being with us right now as we tick off another episode of This Week in Science. Coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to Kirsten and Blair.
Good science to you, Justin, and to Blair, and to all the science minions out there in the world. Welcome to This Week in Science. Thanks so much for showing up on this Wednesday evening to science with us, because we're going to do it. So much ahead, so much ahead. It was a little bit shorter week this week, but still a good week of science. So, on this week's show, I've got some news about life in the stars, the man in the moon, and gut bacteria saved by sugar. Justin, what'd you bring?
I've got guns, semen, teen pregnancy, and maybe we'll save it for the after show, some entanglement hypotheses.
Fancy. So many fancy things. Blair, what do you have?
I have, let's see, dolphins, as previously discussed in the teaser. I also have eyeless fish and an app for bugs.
An app for bugs.
Yeah.
I know a lot of
You
things
heard
in the
me.
digital world don't really make sense, and this is one of them. So I can't wait to hear more about that one. All right, everyone. You guys want the bad news first?
I'll be.
Yeah.
I'll be.
I'm going to depress you first, and then we're going to get to all the fun stuff for the rest of the show. The first case of Ebola has been diagnosed in the United States. Patient began showing symptoms 5 days after returning from West Africa and went to a hospital in Texas for treatment. Blood samples were taken, and then that's where the diagnosis by the CDC was thereafter made. So, it's been said we would find people on U.S. soil with Ebola, with the immensity of the outbreak that has been taking place in Africa. This patient started in West Africa, did not have symptoms while he was on the plane. She, I don't know who the person is. This person was on the plane, was asymptomatic for another 5 days after the flight. So it incubated for a fairly long period of time, which is what Ebola does, what it's been known to do before they actually started showing symptoms. So CDC is not really worried about people who were on the flight. Why are they not concerned? Because Ebola is not an airborne disease. It's not something that if a person has it and they're breathing, they're just going to be infecting people with. It is a disease in which the virus has to be shared via bodily fluids. We've talked about it before. So If you're concerned about whether or not you might have Ebola, ask yourself, have you touched the fluids of anyone with Ebola? If your answer is no, you do not have Ebola.
That's good. Although, what about the airplane bathroom?
Well, the CDC is not concerned because of the asymptomatic nature. And so since there were no symptoms, that means that the virus would not be shedding from the body.
Okay. All right. That's
so good. Yeah.
All right.
So
and what are the
thing.
symptoms of what should I be worried about?
Oh, that's a really good question. I was not prepared for that specific. I'm like, let me get back to the. Okay, I found it. I'm so good, so fast. Thank you, CDC, for being prepared on this aspect of it. Symptoms of Ebola include a fever greater than 101.5, severe headache, muscle pain, weakness, along with that, diarrhea, vomiting, abdominal pain, and unexplained hemorrhage, bleeding or bruising.
And having visited West Africa recently.
And that is the
symptom.
Most important point.
Yeah.
Yes.
So this is important, though. If you visited West Africa and you have a horrible flu, go to the doctor.
Yep.
Actually, maybe call them. Tell them.
Call them
hey,
first and ask them.
I just got back from West Africa and I'm coming down with really flu-like symptoms. Instead of me, walking through the waiting room and having to explain this to people, how about you send me an ambulance and like a couple of plastic bags to wrap up in on my trip.
Yeah.
And maybe get a
good way to do it.
I'll stay in the house till then.
Mm-hmm.
Yeah. But yeah, so it is not time for the United States to close the airports and shipyards, as Whiskey Renegade is saying in our chat room. It is time to start monitoring people who are possibly leaving West Africa for obvious symptoms. It's time to be, to have infrastructure in place, brought there, of course, by international cooperation, but to start placing infrastructure at the gates to minimize the spread of this into other countries. This is probably not going to be the last case that we see here in the United States, but it probably, I'm going to doubt that we will see many cases that are specifically started here in the United States. I doubt we're going to see a lot of spreading here.
I would suggest that number could even be 0.
Yeah. The CDC is on top of this particular case, and I would not question that. Yeah. And as Ed from Connecticut is saying, the good old regular flu is going to kill way more people in the end. And that's true. So maybe think about getting your flu shot, because that at least is currently available. Moving on to some good news.
Mm-hmm.
How about looking for life elsewhere in the stars? You look up at the sky and you think, is there anyone else out there? Anyone?
No.
Possibly? Maybe not.
You never do that, Blair?
Blair's like, nope, I don't do it at all.
Never occurs to me.
Well, a search for the building blocks of amino acids being the building blocks of our DNA, we're looking for the little molecules, complex chains of carbohydrates that create amino acids if they come together and work together. So a search for these building blocks by the Atacama Large Millimeter Submillimeter Array, also known as ALMA, has discovered the most complex molecule to date in dense star-forming regions of space. And so it's a relative of amino acids. It's branched structure, and it was discovered in the Sagittarius B2 cloud, and it's one of the most massive star-forming regions in the Milky Way. It's close to the galaxy's core, 27,000 light years from Earth, and they've seen other organic molecules there, one called normal propyl cyanide, It's the most complex organic molecule found in interstellar space until this one was found, according to Ars Technica. And this one is called isopropyl cyanide. And it has the branch structure is what makes it the most complex that we have seen. So now this finding, basically what it's saying is these dense star-forming regions, these are the places to start to be looking for life, because these are the places where these molecules exist in really high numbers. Numbers high enough that they might be running into each other and running into each other in the proper situations that they could actually, that something could be charged to create life.
The proper conditions, whatever those may happen to be.
Mm-hmm. Yeah.
Sweet.
Yeah. And so so that's the hope. Try and look there. Another thing, looking at the sky, not necessarily looking for life, but when you look up several nights out of the year, out of the month, one specifically, you get the moon with its big face, the man in the moon staring down at you, right? And you wonder, is that man in the moon really made of green cheese?
That one I don't wonder about.
I think about that every night.
That I
Thank
never.
you, Blair. There we go.
But I do see him. I do. I know who you're talking about. I see that guy all the time. Every time I look at the moon, I'm like, there he is. There he is.
There he is. The man in the moon. Well, it turns out that the man in the moon is not made out of green cheese after all. The man in the moon is actually the result of volcanic activity. And the NASA Grail project was recently, or last year, was busy mapping the gravity of the moon. And there were two different, two Grail orbiters orbiting the moon. And based on the gravitational pull that acted on the two of them, they were either pulled closer together or they stretched further apart. And it was this moving closer together and stretching apart that actually enabled a really fine scale resolution of the surface of the moon and its gravitational features, which means how dense the crust is. So they mapped it and they looked at this area that faces us, which is, I didn't know this before, it actually has a name. It's called the Procellarum region. The man in the moon is called the Procellarum region. And it was thought that it had formed from an asteroid impact, that there was some massive impact that had made it into these circular kind of structures.
Mm-hmm.
But when looking at it in this gravitational sense that Grail allowed us to do, turns out that it's not a circle at all. It's not indicative of asteroid impact at all. It's like a polygon. And in fact, when it was mapped, the map was stretched out in a different way. It even looked like a square. So looking at it and the sharp angles of it, researchers went, that is
on the moon.
Yeah, what's this doing on the moon?
What is a square is doing on my moon?
Yeah, what's a square doing on the moon? Why is it so angular? This could not have come from an impact. And so, looking at other aspects of it, they supported the idea that it resulted from volcanic intrusions of magma seeping up beneath the surface and cooling and crystallizing, and that's what the gravity signal from Grail indicates. So, the man in the moon, volcano. Kind of cool, huh?
Mm-hmm. It brings a new meaning to volcano face, huh?
I
had to do it. I'm sorry.
It's true. It could. That's right. That's right. This is no asteroid. It's a volcano. This is This Week in Science. Justin, what do you have?
Let's see. What do you want? Do you want guns, semen, or teen pregnancy?
Don't they all go together?
Yeah.
Well, maybe where you were raised.
Out in the country.
Well, look, let's start here. Let's start with the gun thing. This one's pretty easy. It was a study on gun ownership, and it found that the gun ownership increases the chances of non-stranger firearm homicides. Basically, somebody in the house shooting somebody in the house and them dying. Yeah. New study found that for each 1 percentage point increase in state-level gun ownership, the state's non-stranger homicide rate increased by 0.9%, with firearm homicides increasing by 1.4%. Interesting. It almost goes up then, directly to the percentage of guns. You add 1% guns, you get 1% more non-stranger homicide rate.
Yeah.
Seems like that correlation is pretty strong because it's equal to it.
Interesting. It would be like if there was a recent XKCD cartoon related to statistics that kind of was like this. One person went up and saying, "Oh, it's a good thing I took that statistics class." And the other person says, "Why is that?" He said, "Well, I finally realized that correlation does not imply causation." And they just looked at each other blankly. And that was it. Yeah. Stuff like this, you want to jump to the conclusion and you want to say, yes, look at how causal it seems, but yeah. The reality is that these are real statistics, though, and regardless, the increase is in one place and there is an increase in the other place, and neither of them are good.
So maybe we should just play it safe and have less guns.
Yeah.
It's one of those things about what if it's not causation? What if we accidentally have less guns and less homicides? That would not be a horrible thing.
That would be, scientifically, I think that would be the way that we would be able to get at how it works, at least here in the United States, because we can look at other countries and say, oh, police don't have guns, people don't have that many guns, so, and their homicide rates are lower. So, look at that. You can say what's happening in other countries, and then people say, but that's not the United States. We have different crime problems. We have different stuff going on. And so the only thing that could happen is if we actually did put a ban in place or something to reduce the number of sales and saw sales go down and then saw a reduction. That would be the only thing that we could map that out over time and say these overlay each other. Mm-hmm. It's still observational, though, but that's the closest I think that we could get here in the U.S.
Mm-hmm. Well, I know that in Australia, when they put out a giant gun ban, violent gun crime went down. I want to say it was 97%. It was some enormous number.
That you look at it and you go, wait,
less
what?
Crime. Oh, is that how that works? Less people die when people have less guns.
What about semen, Justin?
Yeah.
What would you like to know about semen?
Well, what was the story that you brought about semen?
Oh, yes.
That was what I was wondering about.
Scientists discovered a form of non-genetic inheritance. Maybe not genetic, non-direct. This is actually showing for the first time that offspring can resemble a mother's previous sexual partner.
What?
In flies.
Elaborate.
This is this confounding idea known as telogamy. Dates back to ancient Greek times, mostly discredited in the early 20th century with the advent of genetics. But their discovery complicates. Here's the quote from Dr. Crean, who's one of the researchers in the project. Our discovery complicates our entire view of how variation is transmitted across generations, but also opens up exciting new possibilities and avenues of research. Just as we think we have things figured out, nature throws us a curveball and shows us how much we still have to learn. Yeah, they say they found that the size of the young was determined by the size of the first male the mother mated with, rather than the second male that actually sired the offspring.
Hmm.
The,
That sounds.
researchers currently propose that the effect is due to molecules in the seminal fluid of the first mate being absorbed by the female's immature eggs and then influencing the growth of offspring of a subsequent mate.
Whoa.
That's interesting. So something that's left over that kind of just sticks around in the female's body long enough to be able to influence the next mating.
Sloppier than you might think it is.
So how recent were these two different mates?
Well, they're flies, so
Right.
I mean,
I know they have short lifespans.
I don't think it was last summer. I think it was probably pretty recent.
Like within the same day?
It had to be that, or it's what flies only live a couple days anyway, right?
Fruit flies, I think they live about a week to 2 weeks.
Okay, it doesn't say. It depends on how long the eggs are there, I suppose. That's the you know, if the previous male's seminal fluid that is affecting them, then they could just, as long as they're there, it's already queued up. Now this is saying affecting size. That the size of the fly might be. It doesn't mean they necessarily are like, oh, that's weird. You've got his nose, but his eyes.
Mm-hmm.
It's not. I don't think it's quite.
But the resulting size of the offspring is going to influence the offspring's mating success.
Right.
So smaller flies, as adults, if this holds into adulthood, smaller flies are probably going to mate less effectively, less often with females. They won't be able to get the mates in the first place. So you are generationally affecting the mating of your competitors, the genes of your competitors.
Yeah.
Downstream effects. Good gracious.
So
what was the
ladies,
other one?
keep this in mind.
Teenage mothers? Is that what it was?
Well, keep this in mind while dating. There's always a chance. There's
Why don't we move along to the next study while you figure that one out?
I have no idea.
Teen pregnancy?
Yeah.
Don't do it.
Yeah, let's talk about something funnier. Teen pregnancy. Oh no, wait. This is actually interesting. This is research by investigators at Washington University School of Medicine, St. Louis, and they were looking at a program called TWIS, basically where they were allowing free contraceptives to go to teens who were already sexually active, who were between the ages of 14 and 17. The national rate of taking long-term contraceptives, IUDs, implants, that sort of thing, nationwide is about 5%. It's pretty small. That most people relying on the pill or condoms and that sort of thing. Those that were allowed to have them free in this program opted for them 72% of the time. So you can imagine that between 2008 and 2013, while this program was running, the rates of pregnancy were dramatically less, and in fact, almost far below the national averages, as were abortions and everything else. So it's sort of an interesting thing because we've had the test case of Tennessee, that the state of Tennessee decided we were going to stop teaching sex education and not be offering it. We'll only teach abstinence, basically.
Right.
This is the only course that we will do in our educational system.
Yeah.
And pregnancy has skyrocketed, and of course, along with that, the abortion rate will go up as well. So yeah, providing free contraception.
Kids will use it.
Mm-hmm.
Teenagers will use it. It doesn't necessarily mean that they're going to have sex more often, which is what the abstinence proponents think. It's like, oh, you're just teaching them it's okay to go ahead and do this. No, it's educating. Educating gives people a choice, whether they're a teenager or an adult. Education leads to knowledge.
Education and then also access, because just educating, you could be doing this if you could afford it. It's not really doesn't really help. It has to be both together. Yeah, in the program, it says the rates of abortions for these teens was 62 to 78% lower than the national rates,
I would say that is success.
depending on ages and that sort of thing. How you were breaking down those statistics. Interestingly to me, that's almost the percentage of the long-term contraceptive percent that was picked up.
Nice.
Good stories, Justin. I think it's about time, though, for...
She loves our creature, bright and small. 5 peg, millipede, no pet at all. If you wanna hear about animals, she's your girl. Except for giant pandas and squirrels, that I'm going for her.
Great. Let's jump right into the deep end with these dolphins, shall we?
Let's do it.
I'm loving the puns this evening. All right. Turns out, dolphins, they have some magnetoreception. So we've talked about magnetoreception with other animals. Specifically, we talked about it a long time ago with pigeons. Since that's been semi-debunked, it's still up for debate. Which animals can and cannot sense magnetic fields is very wiggledy-wobbledy. Science isn't sure. Right? There's lots of animals it seems like they do. We're not sure. We go back and forth. Lots of animals we haven't even tested it with. Dolphins, it turns out, they may be able to sense magnets. So I love this study. It was such a great study. So in Port Saint-Pierre in France, they at the Delphinarium of Planète Sauvage, the Dolphinarium at this place, they tested 6 bottlenose dolphins. So it was a very small sample size, I will say right now. That is a small sample size. However, they had these 6 dolphins. They had them in a bunch of different pools, and then they watched their reaction to barrels. Some of these barrels had strongly magnetized blocks inside, and some of them had demagnetized blocks. Except for that, they were all identical in form, density, shape, everything. So the barrels were indistinguishable. Beyond that, they made sure that the person who was assigned to place the barrels in the pools had no idea whether they were putting magnetized or demagnetized barrels in. So the people couldn't have any error or bias that way. And then
it sounds like it's a properly blinded study, double-blinded study. I like it.
I love when they properly blind a study. They also had the people analyzing the videos have no idea whether the barrels were magnetic or not. So they were just recording dolphin movement. What they found...
At the end of the experiment, they found nobody had been tracking it. Oh, we got to do it all over again.
And they never got research grants ever again. No, so what they found was that the dolphins approached the barrels much faster when they had a magnetized block rather than when they were demagnetized. But the dolphins didn't really interact with the barrel any differently. They just approached it sooner. So as far as they can tell, there's an increased interest or curiosity with the magnetized barrel. But yeah, they didn't really seem to have a preference one way or the other. They didn't treat it any differently. So,
that's awesome.
Yeah.
So this is one of the first bits of really good evidence that dolphins have this kind of sense.
Yeah, so they can tell the difference, and so now a lot more studies need to be done to see maybe why they would need to know a magnetic barrel from a non-magnetic barrel, why they need to be sensitive to magnetic fields, why or how they respond differently to it, because in this case they just ran up to it and it was a barrel. But
Yeah.
so there's some reason that these dolphins can sense magnetism, because we're assuming it's not something that is way, way far down in the family tree of cetaceans. We're assuming that this is something that was evolved, because not every mammal can sense magnetic fields. So why is the question? Why can they tell magnetic fields? What advantage is there? So there's lots of studies now to be done to figure that out.
Yeah. Fascinating. Well, it'll be interesting to know. I mean, there are fish who use electrical, electromagnetic sense. I don't know, navigation or something to do with hunting. I think would be the only, the options I would think would be obvious. But yeah.
Yeah, so that's
Great
their
questions.
number one hypothesis, is that it's related to navigation, but they're just, at this point, they don't have much to go on. So there's definitely going to be more studies now. You know how scientists love to study dolphins, so I'm not concerned. I think we'll figure this one out.
Yeah.
Yeah, the first thing it made me think of is maybe there was something in having a magnetic field there that made it more visible, made the area more visible. So when they were swimming up to it, they're like, "Oh, there's a barrel there. I'll just swim up to that and check it out." Versus the other one, it's like, "Well, I'm gonna have to look at it, wait for my eyes to really see it, and then, kind of come up. Oh, it's just a barrel. Okay." But the other one, somehow it had a better impression of what was there.
Right. Which
Now we got to create a really big dolphin maze. And have some
And use
of it
magnets.
magnetic, and have some of it not, and see if they do better in the magnetic maze than the non-magnetic maze.
Yeah. Well, it's interesting. It could be an element of detection, just kind of figuring out what's around, but dolphins do echolocate. So chances are they could figure out the barrel-shaped object pretty fast. There has to be something about the magnetism that's drawing them to check it out. But I think, yeah, that's definitely what the next step is, to figure out kind of...
Yeah, and also see if a different sample of dolphins has the same effect.
Yes. As I said, 6 is a pretty small sample size. That's enough for a preliminary study to know that more study is needed.
Yeah, exactly.
Yeah, that's pretty much all 6 is good for. Yeah, so speaking of aquatic animals, the eyeless Mexican cavefish. This is a wild animal, and I'll screen share this for those of you watching on the YouTubes. But this guy, he is a looker, I guess.
Translucent-ish? Are those internal organs in that fish I can see?
Yeah, you can
No
see some
eyes.
internal organs, no eyes.
I'm guessing this fish has not seen light. Well, he's blind. 'Cause he doesn't know
Yes,
that. But,
so there's a couple different types of these Mexican cavefish. One of them is a fully eyed fish that lives close to the surface, and then the second is the blind deep water fish. So they were looking at circadian rhythms with these two animals. Circadian rhythms are a change in metabolism over time in relation to lightness and darkness. And if an animal has good circadian rhythms, like we do, even if you are in a dark room, your body will act as though it's daytime, and it will act as though it is nighttime at approximately the correct times. This is why jet lag happens.
Right, our bodies are set to a certain clock, and usually it's light is the stimulus that sets that clock.
Yeah, and they've definitely done studies with animals where they put them in a box for a very long time and their circadian rhythm gets messed up because they haven't had enough light around to know, they get out of sync. But anyway, these guys, so the surface-dwelling fish showed a normal circadian rhythm. Their metabolic rate went up during the day, and that was measured by oxygen consumption. And so then what they were looking at were these two different types of fish, and they were looking at a fixed swimming speed under light and dark and constant dark photo periods, and then they were measuring the oxygen consumption. What they found was that the eyeless cavefish used 30% less energy than the surface-dwelling fish. The surface fish showed circadian rhythms, and they found that the metabolic rhythm persisted even when they were thrown into constant darkness for a little while. But the eyeless cavefish showed no circadian rhythms, and it didn't matter if it was light, if it was dark, if it was light dark. So it looks like the loss of circadian rhythm basically just helps them to save energy. They don't need to amp up for the day. They don't need to calm down at night if they're always in darkness. So actually losing the eyes, so losing the eyes could be for that reason, rather than I don't need these anymore. It's actually a detriment because having eyes means when you are in an area that's light, you're going to fall into the circadian rhythm. So if you're in an area that's most of the time that's dark, maybe not having eyes takes the circadian rhythm away, and it helps you to save energy.
So it's a trade-off between having eyes so that you can see potential predators or potential prey versus saving the energy in a dark environment where you don't necessarily need to see anything like that anyway.
Right, exactly. So this is kind of a chicken-egg scenario. We don't know which came first. We don't know if they were in the darkness and their circadian rhythm was lagging and they lost their eyes, or if they lost their eyes and that's what kind of just brought the circadian rhythm to a screeching halt. We don't know which is which.
But this is crazy because basically every organism down to like yeast that we've looked at has circadian rhythms.
Yeah.
So this is what's weird, is that like we look at cellular organisms, yeast for example, single-celled organism has a circadian rhythm, has some kind of a clock in it that tells it when to ramp things up and when to slow things down, and what is, I guess, the timely time to do things metabolically. And so they're just here's this fish that's gotten rid of it, and this is the first instance we're seeing of this.
Yeah. What I want to know is and of course, yes, classic, Blair's bringing back to mating habits, but I want to know what their mating habits are like, because if it's seasonal, how do they know?
Right.
How do they know?
How would they know?
They would have to. So the males could pick up on hormones from the females, but how are the females cycling properly? Is it something in the water? Is it some sort of pheromones in the water? Is it water temperature? This is what I want to know. Is so much of mating seasonality is based on how much light and dark there is in a day, and how much time has passed since the last mating season, and all of these kinds of things. So, yeah, I'd be very interested to know that side of things for the eyeless Mexican cavefish.
Yeah, I like it. Cool story. I like it. So many questions. That's awesome.
Pretty neat. And then the last one I have is about an app for bugs.
Right.
Yes.
Please explain.
Yes. So insects are really good vectors, as we know. They're really good at transmitting diseases. So this is not an app for the bugs to use. This is an app for humans to use to identify bugs. Aha. So the University of Kansas, computer programmers, scientists, public health officials, and artists all work together to try to make mobile phones able to automatically identify species of disease-carrying insects. This is different than a lot of apps out there. A lot of apps out there, you'll be like, I think this is a Pacific tree frog. And it'll be like, this is what a Pacific tree frog looks like. And you go, yeah, that looks like a Pacific tree frog. And you might even be able to upload a picture, and other people on the internet could say, oh yeah, that's definitely a Pacific tree frog. Or no, there's no way that's a Pacific tree frog. That's actually a Western toad, right? So this is the species identifying apps we have mostly out there now. This app, you would be able to take a picture of this bug, and it will be able to, in an automated system, synthesize the image and tell you what bug it is.
Wow.
Yeah, so this is
amazing.
Awesome. So it's quite complicated, though. You would have to kill the bug, grab the bug, put it in a little box with a backdrop. Mount your iPod on a specific little stand so that you're taking the bug at the exact right angle with the exact right light, with the exact right size, so that the application could look for markers on this bug to be able to positively identify it. And this has all come because of, it's called the Virtual Vector Project, and it's come because of kissing bugs, quote unquote, that are from Mexico, Central America, and South America that spread, and I'm going to say this wrong, Chagas disease?
Chagas.
So that disease is problematic there, and there are certain bugs that spread it and certain bugs that don't, and they can look very similar to the common eye. So they were able to work together.
And if you don't want to, if you're afraid it's the bug that carries Chagas, of course you're going to kill it. So
there you go.
And you can stick it in a box. Right.
And the great thing is, if you identify it, chances are there are more in your house, and then you can step to get your house fumigated and get rid of those bugs. But if it's not one of those bugs, that's an expense you don't have to spend. You don't have to displace your family. That's a big commitment after you see one bug to fumigate your entire house. So it would be really great to be able to say, yes, I need to fumigate my house. This is a danger to my family.
And know definitively that it is or it's not.
Yeah, exactly. I had no idea, but Chagas has infected about 8 million people, 300,000 in the U.S.
Yeah, it's coming. It's one of the diseases. It's called an emerging. What's it called? It's an emerging disease from areas of tropical regions. So moving northward, southward to more temperate regions from the equator. So where Mexico has been experiencing it, it has been spreading northward into Texas, California, things like that.
Do we think this is
south?
I mean, it sounds to me like this could definitely be in relation to a warmer, drier climate.
That's exactly what's going on.
Yeah. So
it's one of the. It's an emerging infectious disease that is the result of a changing climate, and that's what we're seeing a lot of. We're seeing a lot of diseases emerging in this fashion. Chikungunya is another one that I've talked about on the show previously. A little bit scary, and
wait, what is that? What's that one called?
Chikungunya. You gotta look it up. Chikungunya.
I'm not ordering that anymore.
Yeah, but also we're seeing a spread of West Nile. We're seeing a spread of diseases from the East Coast toward the West Coast that have not historically been here. We're seeing the spread of things like bed bugs along the East Coast. There are all sorts of bugs that are starting to spread that are vectors for disease, and they're spreading because the environment is becoming more
bug friendly.
Oh yeah, more bug friendly.
Yeah,
exactly.
Yeah. So these scientists and artists and engineers have worked together so that now you can assemble your bug photo studio in 15 seconds. And they're also working right now to be able to get it to a point where you can 3D print it from anywhere. So if you have a 3D printer or have access to a 3D printer, you can generate one of these photo studios, drop your iPod or your iPhone in there, snap a picture. If you have the app, it'll tell you exactly what it is. And they're hoping to expand it to look at other animals too, or other bugs like ticks, mosquitoes, other things that could be disease vectors to help people recognize what's around them and what is dangerous around them and what isn't, which would be great. I can particularly think about our buddy spiders, who are as bothered as I can be by them. I know that most spiders are not dangerous, and I would be hard-pressed to identify a dangerous spider if I was just kind of seeing one on my wall. And it would be great if you could identify it, and then the next time you see one of those, you can be like, oh, I'll let him go away. It's fine.
I'm not worried about him. He's no problem.
Yeah. Google Black Widow,
and
that's the only one you have to remember.
What about
You're in California.
Don't go to Texas, and that's the only. If you're going to go to Texas, you're going to leave California, if you're going to go to Texas, then you have one more spider to learn.
Mm-hmm.
All other spiders are fine.
Yeah.
Thank you.
We're lucky here. It's pretty good. In Israel, I was pretty nervous about the spiders there.
And on that note, we leave you with spiders while we head to the break because it's time for us to take a little break. Thanks, everyone, for watching so far. We've got gut bugs in mice and a little wetness coming up. Stay tuned. Hey everyone, you know you love TWIS, so this is where I stop and ask you to share a little bit of the love with the rest of the world, share a little bit of your love back with us, all that kind of stuff. We have merchandise you might enjoy, and oh my gosh, it's October already. It's time to start thinking about presents for your favorite TWIS minion-loving people. So who do you love? Who do you know that loves TWIS? Friends, family, buy somebody a little TWIS gift. We've got t-shirts, hats, sweatshirts, tote bags, all sorts of things in our Zazzle store over at TWIS.org. So why not head over there, TWIS.org, look for the Zazzle store in the main header bar, and check out our swag. Give a little love a TWIS for the holidays. Also, we're supported by listeners like you, and your donations pay for everything that we try to do. Pay for things like a new camera that I just bought so that we can go out and do TWIS shorts and make it really look good and get great quality stuff. We appreciate any amount that you're able to give us, $2, $200, $20, whatever. We could not do it without you. You make it possible. And we've made it possible for you to donate in a couple of ways. We have PayPal buttons all over our website at TWIS.org. And so if you like PayPal, you can just go to our website, TWIS.org, and look for the PayPal buttons. They're on basically every page along the side at the bottom of the show notes for every show. And additionally, we have started a Patreon account. And so Patreon's kind of like Kickstarter, where you can support at a certain level per show on an ongoing basis, and you get a little present back from us. So we give you something else. And it's wonderful. It's this wonderful sharing thing. But it's Patreon, patreon.com/thisweekinscience. Patreon.com/thisweekinscience to go to our Patreon page and check out what's going on over there. Whichever you prefer, just head to our website, twist.org, and, keep loving the show. Send us the love. We love it. And if you can't afford a donation, you can't afford swag, I totally understand. We can always use your help spreading the news of TWIS and science. Tell your friends, your family, the coworkers maybe. I don't know. Maybe you know somebody, a bus driver, somebody who just needs to hear more science in their life. Tell them to check out TWIS. Spread the science in the name of TWIS. We thank you for your support, and we really could not do this without you.
And we are back with more This Week in Science.
Oh, yeah. Justin, what else you got there?
Well, here's a question for you. If you were a shark, what kind of personality would you have?
Depends on the type of shark.
Oh, well, that's a tricky question. Yeah. What if you were a Scilurhinus canicula?
Common name, please.
I don't have one. It's a shark found throughout the Northeast Atlantic and Mediterranean. This particular shark, they found that they were, after studying that some were they described them as gregarious, having strong social connections for sharks, where others were much more solitary and preferred to remain inconspicuous in a way, I suppose, away from, some of them were a little bit antisocial. Not that I guess too unusual to find psychological traits in the wild, but I guess this is the first time that they've seen this in sharks.
It looks like they're called spotted cat sharks.
Spotted cat shark.
Mm.
Now, we found that even though the sizes of the groups forming changed, socially well-connected individuals remained well-connected under each new habitat. In other words, their social network positions were repeated throughout time and across different habitats.
So it's pretty consistent in a particular shark, and they wouldn't. I wonder if a shark would have good days or bad days. A moody shark.
What's interesting too is I guess these have somewhat of ability to create camouflage. It says that the less social individuals tended to camouflage themselves alone, matching their skin color with the color of the gravel substrate at the bottom of the tank in which they were being observed.
That's cool.
I think it makes sense that sharks could be moody just like any other animal.
Oh, I think it makes absolute sense. I mean, we find the more we look at different animals in ways that we search for these kinds of humanistic traits, what we think is human, the more we find that those traits are there, and we're seeing it more and more in fish. And that's, I think, the thing that people find so surprising. They're like, fish are just slimy. Fish flop around when you take them out of the water. Fish taste good. But we're finding out they can feel pain. They have personalities.
Yeah.
If you think about if you've had fish as a pet, as a kid, there's definitely differences from one fish to another, even in goldfish, you know. Some, especially if you really try to interact with the fish, some of them will come right up when you feed them. Some of them, if you stick your finger in there, will want to get touched, and others won't. I think that there's definitely, among any animal that lives around other members of the same species, they're going to have social dynamics, right? That just seems like an intrinsic part of that.
And social dynamics, you would need some amount of personality for social dynamics.
Mm-hmm.
Yeah.
I saw a, what are they called, the African great horned bird? I can't remember it's like an Assyrian something or another. Anyway, we were at the zoo the other day, and I swear this bird had a little mouse in its beak, a little dead mouse that was obviously a feeder mouse, and was parading it as we walked up and down in front of us with the cage, holding it up, kind of showing it off.
Look what I got.
Totally, right? And as soon as we left, I looked back, and it was just like, oh, well, like done. But the whole time we were there, it's like, check it out. I got one, and you don't. Totally showing it off.
Nice.
Yeah, I had a friend with a pufferfish for a while in her aquarium, and it was a very friendly pufferfish.
Really?
It really liked attention. If you were in the room, it didn't matter if you were just feeding. It wanted your attention. It wanted you to actually pet it. So it would come up and try and get that attention. Wow. It was pretty cool.
So funny.
From that point forward, I was like, I think pufferfish are the best fish ever. Oh my gosh.
They're pretty cute. Their little beak. Yeah.
So cute. Speaking of fish and sharks, they live in the water, right? So they probably, I wonder if they know what it feels like to feel wetness, to feel wet, right? We know what it is to feel wet. Yeah. You know when you touch water.
Right. And we can feel varying degrees of dryness. But I don't know if you're in the water. Yeah, do you feel varying degrees of wet?
All the time.
It's a little extra moist today. There's a lot of humidity in the water.
Yeah, so researchers actually, there's a study out recently in I think it's Journal of Neuroscience. Yep, Neurophysiology, Journal of Neurophysiology. Researchers looking at what our brains are taking in to tell us so that we perceive wet. Because we don't have special receptors in our skin for wetness. There's no special nerve fiber that's like, this is the wet fiber. And when your finger gets wet, that goes bzz, 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 and suddenly you know you're wet because that's the signal. There's no bat signal like that. So how does our brain know? And what they did some experiments and created one of the first models that people have tried to come up with to figure it out. And what they determined is that it actually has a lot to do with temperature.
Yeah, that makes sense.
So colder temperatures are actually more likely to be perceived as wet than warmer temperatures. And there's a perception, and it's kind of wetness is they've quoted it as a perceptual illusion, really, because we don't have a direct signal from the skin. We also have nerve fibers, these alpha nerves, that run through the skin, and they transmit tactile and pressure information and temperature information. So part of their experiment, they actually blocked the alpha nerve fibers to see what would change people's perception of feeling wet, and they found that wet perception increased as temperature decreased. They found that they're less sensitive to wetness when the A nerve fibers, the alpha nerve fibers, were blocked. And the skin that has hair on it, hairy skin, is more sensitive to wet than your fingertips, glabrous skin that doesn't have any hair in it. So what happens is you feel wetness based on a combination of the touch of water on your skin and how the nerve fibers are stimulated based on the tactile sensation, and then additionally with the combined information of temperature. And so I think very often what's going to happen is that like colder water will probably wick heat away from the skin and start to make you feel colder, damp.
Yeah, your skin temperature would actually change because it's transferring heat to the water.
Yes. So it's heat transfer.
But what about when you get in a warm tub?
Right. So these are questions. Right. Is there a sound quality to it? They didn't include anything other than this extremity-based test. So temperature and blocking the nerve fibers on people's arms. But what about other stuff? Is there another stimuli that they're not counting in there somehow?
Well, what I think about is we can tell textures, right? We can tell smooth from rough. So I think it has to do with smoothness and viscosity.
Viscosity may be very important, how it sticks to the skin as it goes past, because water has a definite surface tension. And so there could be something that we learn as we come to use water as we grow up, that we learn is the surface tension experience, the viscosity experience of water.
Yeah. That's interesting.
Yeah. So, it's a really interesting question. If you really think about it, what is wet? How does our brain know what is wet? This is we don't know.
My brain hurts now.
Right? Yeah. Anyway, that's cool stuff. Let's move along. Justin, you have another one?
No, that was it for me.
All right, Blair, did you have some more?
Sure. Let's see. Oh, there's a new method to motivate students to reduce energy consumption. And basically, it's to shame them in real time. So this research from the UK found that a combination of real-time feedback together with a human energy delegate they did this in 8 halls of residence in a boarding school. So having a delegate in charge of students and then having everyone know in real time how much energy they're using at all times resulted in a reduction of 37% in energy consumption.
Wow.
That's enormous, 37% reduction. And that in the opposite, the other side of things, in 8 different halls, they had these students exposed to real-time feedback and a weekly email alert. So they weren't seeking out the feedback as much, and they didn't have specific people in charge of them. They weren't being publicly pointed out for doing good or bad. and they only saw a 3.5% reduction in energy consumption. So this is another indication that it is community-based solutions is what's going to change how we do things. Doing things together as a group, us talking about it, having a dialogue, that's what's going to make this happen. That's why people
Social pressure.
Exactly. That's why people started recycling. That's why there was a big Prius thing that happened, is because it was something that was out in front of everybody. That's why green efforts have become such a big thing, and green is such a hip word because it's hip to be green now. So if you can brag to somebody, that is, or you can be shamed for having your plastic bag out or not bringing your reusable bag to the store, is happening now in San Francisco.
Mm-hmm.
That's a huge component. We're social creatures. We're just talking about gregarious natures and animals. We are a gregarious species. We're social. So if we tap into the social structure and the social normalcy of saving energy, using renewable energy, all of these things, that's what's going to make the change. That's what it looks like. And related to that, by the way, the governor signed the plastic bag ban yesterday for all of California.
All of California.
Yeah.
Nice.
Yeah, we gotta. I don't know what the timetable is for the rollout exactly yet.
July 2015.
There it is. Mm-hmm. So yeah, as of July 2015, you will no longer be able to simply go to the next city over if you're in Davis and get triple bagged and then come home.
Yep.
Triple plastic bag.
Bags, man. Start carrying around your bags.
There will still be paper bags, but it starts making people think about it more when the person behind the counter at cash register looks at you and gives you that glare.
Do you need a bag today? Okay, 10 cents, please. So I thought about it, though, and it hasn't even been 2 years, or maybe it's been just about 2 years, since the plastic bag ban in San Francisco. And we've come a long way. It was hard at first. It was definitely hard. But in just 2 years, pretty much everybody has adjusted to that way of life here. And
I have learned that I am terrible at remembering to put bags back in my car. Yeah. That's what I have learned.
Yeah.
But I recycle those paper bags like mad.
There you go. There you go.
You know, so here's my method. Actually, I did it even today. Because there's a step to the process I always forget, which is when I go to the car to bring all the little tote bags with me and put it in the car.
That's what I forget.
Because that's where it gets lost. Because I'm not always going directly to the store. It's like, you know what? I got enough time here. Why don't I go do the shopping? I'll go knock that off the list. So what ends up happening is I go there. The totes are in the kitchen where they were unloaded of groceries from the last time. So then I get everything. No, I don't want the bag. Just put everything loose in the cart. Just put it from the counter into the cart.
I do that too.
Then I go to the car. I load up the back of the Prius with the groceries. Then I go home. Then I go into the kitchen. I grab the totes. I bring them out to the car. That's when the bagging happens. And it only happens to get groceries from the car into the kitchen.
You know what? It works. Less plastic in our environment is a good thing. Less emissions from making the plastic. Mm-hmm. Less plastic in our oceans. Less plastic in our landfill. It's a win-win-win.
Yep.
Sorry, guys. It's going to be a rough transition, but in the long run, we will be happy we did it. And where California goes, hopefully the rest of the country won't be that far to follow, because California is, the 4th or the 5th largest economy in the world. So if we can do it, anybody can do it.
So am I. Woohoo!
Yeah.
Gut bacteria and sugar. But it's not.
Oh, I love this story. This is interesting.
This is super interesting. It's not the kind of sugar you think. I'm not saying that they gave mice a bunch of sugar and it saved their gut bacteria. What happened in this study is that researchers found that sick mice that they infected with something and made them sick, so that they were their tummies were upset and they didn't want to eat. They weren't feeling good, so they weren't eating a lot of calories. Normally, you eat a lot. You eat calories. Your food goes in. The bacteria in your small intestine love to help eat that up too, and so you're helping to keep your bacteria alive by eating. But if you get sick and you stop eating, okay, that might kill off the bad bacteria, but it's going to kill the good bacteria too. So what's going on? How are we protecting our good bacteria when we get sick? Is our body doing that? Are we trying to keep the good bacteria in while our body gets rid of bad stuff? Yes. In mice, at least, that's what's going on. They found that sick mice produce this special sugar. It's not a normal sugar. It's called L-fucose. Not fructose, L-fucose. And L-fucose can't be used by the mouse. So the mouse doesn't suddenly digest it when it's been produced and have that become body energy for the body that would then feed all sorts of stuff. So it's the host doesn't use it, the mouse doesn't use it, but then when it binds with proteins, it can be used by microbes as a food source. Normally, the small intestine of mice doesn't produce any L-fucose, like just very little. It's a very low-level sugar. It doesn't really exist in there. What they found is that the L-fucose gets produced in the small intestine during illness. They made mice, they genetically engineered them to lack the gene that is responsible for the production of this sugar. And they found that under normal conditions, when the mice were healthy, those without the gene for the L-fucose, they regained weight after an induced sickness much slower than their normal counterparts. But the ones with the intact gut microbiota and the ability to produce it, the sugar, recovered efficiently. And so mice that can produce this sugar recover better from illness than those that can't. And so if you remove bacteria, the effect goes away. And so basically what's going on is that this L-fucose is involved in supporting the microbiota. And the interesting thing to this that really has a link to human health is that about 20% of humans lack a functional gene to create L-fucose. Those 20%, or not, I'm not going to say it's specifically linked between the 20%, but a lack of an ability to produce L-fucose has been associated with Crohn's disease. And so the question now is whether or not this gene we can do something with the gene or create something that can help people with this gut ailment that will actually help to keep their gut bacteria healthy, so that when they get sick, they won't have they won't lose their good gut bacteria, and they'll be able to fight off infection, and they won't have strange gut inflammation that leads to all sorts of weird microbiota problems later.
Nice.
Yeah.
So great. Feeding our little bacterial pets. Huh?
Next time you're sick, eat cookies.
Next time you're sick.
That's not exactly what.
They're not exactly.
All right.
Am I taking one piece of information from a story incorrectly, applying it to the macro world?
Yeah.
Yes.
Here's one also related to gut bacteria. Apples could make you skinny. Specifically, Granny Smith apples are very good at promoting beneficial gut bacteria and have been linked with a reduction in obesity, eating Granny Smith apples and having these beneficial gut bacteria. The Granny Smith have the highest of all the apples of non-digestible fibers in them, and those ferment in your gut and help that friendly bacteria grow.
Yeah,
So.
I was just reading a story today, it's not a study that has happened, but there's an ongoing study into these tribal people called the Hadza, who have what is considered the closest to our ancestors' gut microbiota. because of the way they live, where they live in South Africa, and the food they eat and everything. And one of the things that was talked about in this story is that they get fiber every day. They eat these tubers and these fruits, no matter the season. So they have a really good fiber diet every day, and they have all these bacteria that don't exist in the Western gut, that are almost completely wiped out in the Western gut. And so one of the questions is whether fiber has something to do with that.
Fiber is a good thing. Eat your fiber, everyone.
Yeah. And finally, if you're looking to the sky again, there are some really wonderful images that have been shared by the Maven. And let's see, the Maven and the Mom. Maven and Mom. Here, I just shared the wrong screen, but the Maven and Mom missions to Mars. Maven is NASA's, and I believe, and Mom is from India. And here we have a picture of the red planet from Mom. Mom is taking pictures in the visible light spectrum, and so we can expect to see true red colors of the nice red dusty planet. In this image, you can see there's that I'm showing on the screen currently. There's kind of a dust storm brewing on one of the poles of Mars, and then an additional image from Mom was taken showing the layer of the thin atmosphere that Mars has over the planet. Nice. You can kind of see through there's a little bit of translucency through the thin gases that cover the planet. And then Maven is taking pictures not so much in the visible spectrum. Maven is looking at Mars in completely different colors. And so some of the first images that have come back from Maven are looking at the composite molecules, the elements that are held within the atmosphere. And so one of the images is an amazing bright blue that shows the hydrogen in its atmosphere and how that extends around the surface of the planet. The green image shows oxygen, which is very close to the planet's surface and does not extend very far out. And then there's also reflected sunlight and composite imagery. And so you can expect from the Mom images to get kind of more of a true to life, what does the planet look like view. And then Maven is going to show us a lot more in-depth stuff that our eyes cannot normally see.
Nice.
Yeah.
Beautiful.
Yeah, so these things ended up in the sky around Mars, a couple weeks ago? Really not that long ago at all. And they're already sending back great imagery, so it's pretty cool. Things happen fast these days. Oh my goodness. Are you ready to end this show?
Let's do it.
All right, everyone. I would like to give shout-outs to the Patreon sponsors who support us here doing our This Week in Sciencing every week. Thank you so much to Brian Condren, Kevin, Patrick Cohn, Kevin Donald, Wesley Ballard, Paul D. Disney, Sam Winzier, John Ratnaswamy, Rudy Garcia, Aubrey Burgess, Stephen B., Jason Dozier, Matthew Litwin, Eric Knapp, Andreas Akestad, Jason Roberts, Shane and Tara Ginsburg, Byron Lee, E.O., Bob Calder, Jared Lysette, Ulysses Adkins, Jake Jones, Cassie Lester, Chris Cross, Sarah Chavez, Stephen Soroyech, Layla Amir Sadeghi, Paul Stanton, Marshall Clark, Charlene Davidson Henry, Violated Gorilla, Don Kamarechka, Randy Mazuka, Ed Dyer, and Tony Steele. Lord Trentonian Moody, Larry Garcia, Richard Bowersox, Kevin Parachan, Craig Landon, sorry about that, Russell Jensen, Mountain Sloth, Jim Drapeau, Darren Lute, Tara Payne, Alex Wilson, Daryl Lambert, John Maloney, Jason Olds, Gordon Grant, James, Paul West, Lauren Lang, Ed Bennett, Thomas Mikinen, Alec Doty, Illumin Lama, Nick Gradwell, Dan Rambo, Dougal Campbell, TMRO, Miko Pakola, Craig Porter, Adam Mishkan, Jason Martin, Marjorie, Scott Lajewski, Tyler Harrison, Ben Rothig, Columbo Ahmed, and Gary Swinsberg. Thank you so much for your support of This Week in Science and all that we try to do. Those of you who would like to support us, you can find our Patreon account at patreon.com/thisweekinscience. Next week, we'll be back on Wednesday night for the rest of October. Wednesday nights, we're broadcasting live, 8 p.m. Pacific time. on twis.org/live. But don't worry if you can't catch us on a Wednesday night, or maybe this is your first time you can catch us live because it is a Wednesday night. Whatever. We do also have our podcast at twis.org. You can subscribe there too. And we also have our YouTube page, which is youtube.com/user/thisweekinscience. Tell a friend about TWIS.
Yeah, thank you for enjoying the show. We hope you join us again next week at the same time. TWIS will also be available as the podcast. Just search for This Week in Science in your iTunes directory. Or if you have a mobile device, you can Google us there. TWIS for Droid app in the Android marketplace, or TWIS, TWIS, in the Apple marketplace, though we do have to do a drill down to the new iPhone podcasting app, which is bringing up This Week in Scripture instead of...
Oh, no.
Oh,
Something.
That's not ideal.
that's not ideal.
Something must be done.
Well, for more information on anything you've heard here today, show notes will be available on our website. That's www.twis.org. That's TWIS.org. There you can also make comments and start conversations with the hosts and other listeners.
Or you can contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistmeaning@gmail.com, or Blair at blairbaz@twis.org. Just be sure to put TWIS, TWIS, somewhere in the subject line, or your email will be spam filtered into oblivion. You can also hit us up on the Twitter. We are at TWIS Science, at Dr. Kiki, at JacksonFly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that came to you in the night, please let us know.
We'll be back here next week on Wednesday, and we hope you'll join us again for some more great science news.
And if you have learned anything from tonight's show, remember,
It's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news. That what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just better understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got a laundry list of items I want to address. From stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say. And if you learned anything from the words that we said, then please just remember it's all in your head. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.
All right, Kiki, we need to get a physicist on the show. Stat.
Okay, that sounds good. Let's do it.
I need like a Sean Carroll.
I could probably get in touch with Sean and see if he'd want to.
Yeah, mostly I just want to spring one question on because there's something that's got me completely puzzled now, and I think the answer seems obvious to me, but it seems like something's not right. So this quantum entanglement thing last week, right? Got the entangled particle. One goes into a crystal. One goes down a long thing. You hit the one 25 kilometers away.
Right.
The photon.
Yes. The 25-kilometer away thing.
The photon emits in the crystal. Now, this is at different distances, and obviously the distances don't matter. But why is this so spooky? Isn't the answer to this that from the perspective of the light, the entangled particle, the light, that those things happen at the same time, right? Because light doesn't experience time. It seems like sort of like a parlor trick of like, yes, this is dilated time. This is an extreme example where you're looking at dilated time of light versus our meet spacey, slow progression through tick-tock time. Because, you know, if you put this an electron in a super collider and just spin it around, and then you have the magic ability to have the super collider ask the super collider, where's the electron, without it actually interfering with it, without doing a detection. It would say, I don't see an electron, I see a circle. There's a circle there. And the circle would go, yeah, that's me. I am a circle, the electron. I am the circle. Because it's everywhere at once. It's not experiencing time. It's in all of those places around the circle at the same time. If the entangled system is this, entangled and it splits off, the photon that you hit it with hits, and from its perspective, from the light's perspective, at the same moment, same instance, 25 kilometers away from the moment it was entangled, that happened at the same time to it. So it's emitting the photon over here, but over there, but it hasn't gone anywhere in time at all. It hasn't experienced any time.
Right.
That should just be what that is. And maybe that is. Maybe that is the answer. I don't know. But I need to ask, like a Sean Carroll to clarify if that's just what it is. If it's just from the perspective of the light, there's no difference in location there or time. It's the same thing.
So, yeah, thinking of it of perspective from the particles. Yeah, I don't know. That's interesting.
Yeah.
I cannot answer this question.
That's what Einstein's always saying. Einstein's always saying light doesn't experience time. It's not moving in time.
Right.
It's standing still in time. If that's true, then just because our location of the particle isn't defined until we hit it, its time is also undefined. It's throughout all of its time, and it's probably throughout all its time between interactions too. Like I sort of picture the tick-tock of light time being from one interaction to the next. Those are the only points. So maybe it's the point of entanglement to the point where it's hit by the other photon, or it's put into a detector of some sort to check its upspin, downspin. Those two ticks of its existence happened at the same time from its perspective. There's no difference. They're basically instantaneous. So our whole moving something that's time dilated, kilometers away, right, absolutely changes nothing if it's experiencing all of its time at the same moment.
Right.
Cool.
I don't know.
I fell down an iTunes hole. I'm trying to figure out. Yeah, if you search TWIS in the podcast app, we don't come up at all.
In the App Store?
In the you know how there's a podcast app now? So you don't go to iTunes, search for This Week in Science and download it. Now you subscribe to podcasts via the podcast app on the iPhone, and we don't come up in there at all under TWIS. You have to search This Week in Science.
Okay.
So I don't know how to fix that. But.
And we had TWIS, and in this case, this is beautiful. There was This Week in Science before there was Scripture.
Yeah.
Yeah. Well, also This Week in Salsa came up too.
which I actually subscribe to. I found it by accident once. I actually do subscribe to This Week in Salsa. It's phenomenal.
So if somebody can tell me how to change the iTunes information so that TWIS is something that comes up and is part of the search criteria, I can see about doing that. I don't have an iPhone, so I have absolutely no idea what you're talking about.
Slightly third-party app that's not just taking from iTunes. It might have nothing to do with iTunes selectivity or anything you change in iTunes. You might have to like go to the podcast app and be like, "Hey, we're a podcast that you should have in
Right.
there."
Is this something that I should have subscribed to that I didn't know about because I don't have an iPhone?
Because it's new. It's new. They just started doing it.
Yeah. I don't know. Yeah, if anyone can send me information on that, and then, yeah, these are the kinds of things that hurt my brain. Trying to keep up with all this technology. Got other things to do.
Oh, what do you call a caveman that's lost and looking for its home? A Neanderthal.
Nice one. Was that Frogs Go Berserk Over Worm video that Janiscu put up there? Is that the one where the iPad or the iPhone or whatever is put up and then frogs are like attacking the screen? I've seen that one before. It's pretty funny.
That's fine.
Yeah, and even though on Google Plus, even though I do the This Week in Science TWIS, you still can't. It's hard to find us via TWIS also in Google Plus. And I don't know these people who are supposed to be good at search engines. Why don't they make it so they work the way I want them to? Yeah, I think I need to find somebody. It might be
easier for us to learn the way that they've created the system.
Yeah, but everybody keeps creating new systems, and it's hard to keep up. And so what I need to do is find somebody who can consult on that level for us, because I don't know. I need help. I need help doing this stuff. I need help. Anyway, it's 9:30, and
let's go.
I'm sorry, everyone who's here for a long after show, but I'm gonna bail.
I have to bail too.
I have a 7 a.m. flight tomorrow morning. I'm going to Alabama. I'll be at the University of Alabama, Birmingham, on Friday. Well, tomorrow I get there, but then I'm there on Friday.
Are you gonna talk like that the whole time you're there?
No.
Alabama.
What's up,
If I do, people need to throw things at me, right?
Nobody talks like that here, sir.
I'll be in Alabama. So I have to get up early for my flight to be able to get on the flight so that I make the flight. Because if I miss the flight, that means I miss 2 flights, which is harder to make up if you miss connecting flights with connecting flights. So I have to do this. And then... yeah, I just have to get up and do that. And then since I'm going to be gone doing this, if anybody is going to be expecting the podcast version of this to come out, it probably will not come out until sometime Monday, because that's when I will be back at work again. So I will not be editing the show until Monday, unless of course I get a massive amount of time tomorrow, and I have an ability to download on my laptop and all that kind of stuff, which I'm not counting.
What's an Elo? All the kids are on Elo now.
I know, all the kids are on Elo. Thanks, Identity4. Yeah, Ello's the new social network that people are talking about, and I don't know. I haven't gone.
I
I've
don't know
got
what
an
that is.
invitation. I've gotten an invitation or two, but I haven't joined it yet.
Come on, Kiki. We can't be the last ones on Face Space or My Book or whatever this thing's called. We got to move on. We got to keep going.
Oh, I know.
Hey,
I
it's
know. I
time.
need to do
Come on.
this. I know. I
Gotta do
know.
whatever
It's
them
supposed to
young
be. It's supposed to be the
whippersnappers are doing.
Facebook with no advertising, except
Yeah.
it's nothing like Facebook, and probably doesn't act like Facebook either. And it's very low tech, and I don't know. I haven't heard anything really impressive yet. So, but yes, we need to be on it because it's a social network, and so we need to be on it. Which is also why we need to have
Ugh.
an Instagram, and why we have to have, I don't know, other.
Oh yeah, we don't have an Instagram, huh?
You know, we need
We
an
don't?
Instagram. I personally don't have an
I'm getting one
Instagram.
of the.
I have an Instagram.
I'm getting one of the. My old phone died. Pretty much done. I can basically do text messages on it now, and receive parts of phone calls. So I've got the new phone on order.
Awesome.
And apparently that one comes with the Pinterest thing.
So I don't know if you can manage multiple Instagram accounts from one phone, but I could certainly start an Instagram account. I can look into it.
Okay.
Oh, I'll look into it.
But then don't we use those accounts just
Yeah,
to
if we
feed
could-
stuff
if-
into the accounts that we already have in other places that where the people actually are?
Right.
I mean, can we all agree on one?
Can we all just agree to be in one place?
I would like that.
Like make one that's not like a corporate one, but one that's like this is just the public forum one.
Oh,
This is
Gord
all-
McLeod likes Ello.
It hurt my head when I just looked at it.
Yeah. More like
Of course,
Twitter
my
than
solution
Facebook.
for narrowing down the number of social networks that are out there is to create yet another one.
Exactly. I've always loved Twitter, and it's always been the place that I've loved to be the most.
Twitter?
It's always what I've loved the most. I like Twitter. That's the one I like.
I should look at my Twitter. I don't look at my Twitter enough. I have one somewhere.
It's the one that's not making
I
a profit
misplaced it.
either. Anyway, Identity 4, to answer your question, I don't know that you can help on editing because I really appreciate the offer. That would be amazing. But yeah, I just need to. I do certain things to it. I end up putting it, in the right format with all the tags and ID3 tags and keywords and everything, and this process that I go through, and it's difficult.
Why don't we
to
create?
pass
This is
off
what we
in
should
a
do.
way.
Instead of spending all this time on this dang radio show thing, do something to make some money. Why
Right?
don't we create a social network for
Yeah.
science-minded people and call it, I don't know, Science Island,
I like it.
right? And
Let's
then we
do
can
it.
all virtually hang out, share sciencey news stories, newest science evidence. Just people who like science. Everybody else, just take a hike.
Oh my gosh, we're so close to our first milestone on Patreon.
I know, we're so close. We're up at $480, everyone. We're almost to our first milestone. Woohoo! This is the beginning of
Wait, I thought we already had a milestone. Wasn't it like
No.
We're doing awesome. We're doing great. Everyone, thank you so much. Thanks, Identity 4. Thank you, everybody. I gotta go finish packing and get some sleep. Blair has to pack. We've got some traveling
Wait,
to
where's
do.
Blair? Where are you going?
I'm going to Las Vegas.
Las Vegas?
Yeah.
Yeah, and maybe
Alright.
someday I can teach a minion to fill in. It's true, because it would be nice to keep getting the podcast out on schedule and all that kind of stuff.
Yeah.
We need an intern.
Yeah, we need an intern. We need a media intern, marketing intern is what we need.
Yeah.
We need an agent. You know what we really need? We need our own.
What we really need is for me to go to sleep right now.
Yeah. All right. Goodbye.
Thank you so much, everyone, for watching the show. Really appreciate you being here. We switched to Wednesdays, and you made it.
Mhm.
Thank you for watching and being here. And those of you who are watching after the fact, thanks for watching and being here. Maybe we'll make it next Wednesday.
Also, should we somebody should go tomorrow at the regular time into the chat room. Should show up in the chat room just as an explanation in case they missed the message.
Why don't you try that?
Yeah, why don't you do that, Justin? Because I will.
I'm gonna be at work. This was the whole problem.
I'm not gonna be here either.
I'm gonna be at Presidio Teachers Night. Sorry.
This is why Thursdays was bad.
Wait, you're gonna be in Las Vegas.
No. Starting Friday, I'll be in Las Vegas.
Hey, you guys,
You're packing for somewhere you don't have to be tomorrow.
I'm working a 12-hour day and leaving at 7 a.m. on Friday. I have to pack now.
This is all tomorrow. This is tomorrow, Blair. This is the Blair that never does anything for you. She never plans ahead for you, right?
This is
nothing. You have to put in all the work.
Talking to my 3-year-old.
She just gets to sleep, be rested, whatever.
I'm going to count to 3.
Did yesterday Blair get enough sleep?
I haven't stopped by the time I count to 3. I'm just hanging up. Oh, I just get to do it. 1, 2, 3. Good night, everyone.